We are starting week 5, and because this is an exam week, I won't be holding our normal Zoom on Tuesday. So I've pre-recorded the Zoom, and that way you can concentrate on doing your exam on Tuesday. So the exam I went over in the week 4 Zoom. So if you haven't done the exam and you're looking for that information, the exam review is in the week 4 Zoom, which is posted under weekly instructions under week 4, and it's on our YouTube channel. So I'm going to look at the other things that we have coming up in the next several weeks. So we've got not this week, but in week 6, you've got your first full lab report due on October 10th, which is why I've just sort of been pushing you to get your labs done so you can choose one of those labs for the lab report. When you get that graded, you will have a chance to revise it and submit it for some additional points. And then in week 7, on October 17th, you have a midterm review of your scientific paper blog, where you're just basically submitting the link to the paper and the link to the blog. And you're going to look at other people's blogs the following week, and they're going to look at your blog and give you feedback on it. And we know they're not completely done, but they should be about halfway done at that point. So October 17th is when you have to have the links to the blog submitted. And then October 24th is the date you'd have to have your reviews done, which would be week 8. And then on October 31st, the end of the month of October, you're going to have your second lecture module due. So you've already finished the first lecture module. Now you're going to be working on the next 4 chapters and 4 investigating the evidence. And this one, instead of having 1 homework assignment, it has 3 homework assignments, like the biome assignment you had in lecture module number 1. And then Tuesday of this week, you've got exam 1. On November 2nd, you have exam 2. So this week, we've got the Mark Recapture Lab. Next week, you're going to be doing the Island Biogeography Lab, then the Dispersion Lab, and then finally the Survivorship Lab. There we go. So week 5. After you finished your exam, then you can start looking at lecture module number 2, which we're going to be starting with the next chapter in the book. So that will be chapter 7. So we're still dealing with how the individual interacts with the environment. And this is looking at energy and nutrients. You'll also be doing investigating the evidence number 4. And then this week, your lab is the Mark Recapture Lab, which is a very straightforward lab. So I'm going to go over that one. And then you should be working on your first full lab report. So even though it's not due until week 6, October 10th, you should start working on it this week. So pick the lab that you're going to write up for your lab report. You're going to be having to use all the class data for your lab, or at least the class data that's relevant to your hypothesis. You come up with your hypothesis. You're going to have to write it and proofread it, and then you're going to submit it. So start working on that this week. And then this week on your scientific paper blog, you're going to put information about the hypothesis that they're testing in your paper and why they're studying it. And if they've got a difficult vocabulary that you're running across, start putting that together as well and explain any necessary background information. So chapter 7, I'm just going to go over that. We've got the videos for chapter 7. So the first video is the first section of the chapter, which is on energy and autotrophs. So you should remember how photosynthesis works and cellular respiration work. And then we talk about chemosynthesis. So this little 3-minute video is from the BBC on volcanic springs and hydrothermal vents. Then the next part of the chapter, so we're talking about autotrophs, so photosynthesis for the first part. Then we're going to talk about heterotrophs, so you're going to be talking about things that eat other things or consumers. So that's a 35-minute video. And then talk about how we are limited and how much energy we have. So we're talking about that principle of allocation and how we allocate energy and how that applies to looking for food. So we also talk about optimal foraging and optimal foraging theory. And then there's two little one little video here on some heterotrophs and looking at their foraging, and that would be looking at this little video of cheetah cubs learning how to hunt. And then this video, ecosystem ecology, links in the chain. So this is from an ecology video series that really summarizes a lot of the stuff we talk about in this particular chapter, and that might be good for you to review when you're coming down to reviewing for exam number 2. So that's an optional video, but you might find it's a good overview and maybe a different perspective on things. So that is only an 11-minute video. And then the Mark Recapture Lab, the how to collect the data and analyze the data are all in one video this time instead of separated in 2. So it's a 34-minute video. So watch this before you do the lab, and then you can go back and reopen it when you're going to do the analysis. And a fun little video on doing Mark Recapture, estimating how many beer they drank in an evening, was using beer caps. And so you can, that's an optional one, but it's a nice little 7-minute video. So the chapter, we start off with autotrophs and photosynthesis, and then chemosynthesis for the first video. The second video, we're looking at energy and nutrients and how we get adequate supplies of both. And then we talk about energy limitation, optimal foraging, which is the last 2 sections of chapter 7. When you're going through the chapters, these are the concept review questions you should be doing for each section. So I didn't ask any for section 7.2. So 7.1, the 3 questions, 7.3, the 3 questions, same for 7.4 and 7.5. And then the review questions at the end of the chapter, I think you should be able to answer for an exam are 2, 3, 4, 5, 7, and 9. And then you've got the investigating the evidence on variance. And this variance relates to a coming up lab that you're going to be doing. So you're going to should be able to answer questions 1 and 2 on that. So the lab we're doing this week is the Mark Recapture Lab, which is a very common technique, and there's a number of different variations on the technique used for actually estimating the population sizes of animals, things that are walking around, things that might be trying to avoid you, things that might be hard to tell apart. So you've done looking at populations of plants, you've looked at pollinators. Now we're going to be doing another population estimate using Mark Recapture. And so it's a standardized method for capturing and observing animals. And you will see if you're looking at any papers for the scientific paper reviews that are dealing with population sizes of animals, the chances are very good they did some sort of Mark Recapture technique. And if you look at like this picture of the penguins here, if you're trying to count the number of the penguins, and then you start to count them and then they start shuffling around and you don't know which penguin you'd already seen before, obviously marking a penguin is going to be helpful so you know what ones have been counted and what ones haven't. So in order to do this lab, you're not going to be using penguins. You're going to be using the box of toothpicks that come in your kit. So you have a box of 500 toothpicks, so we know the population size. You're going to scatter all 500 toothpicks. Don't scatter 20, scatter all 5. Don't do it in a place where you've got really long grass or a lot of leaves are covering the grass. Make sure a park is a good place. There's good places on campus where we have lawns, and you're going to scatter your 500 toothpicks. The best thing to do is get someone to come with you and they can scatter the toothpicks. And then your first step after they've scattered them is you're going to go out and you're going to, you've got it timed, you've got all the instructions in e-learning and in your lab book. You're going to capture as many of your individuals as you can find, as many of your toothpicks as you can find, and you're going to mark them. You're going to count them up and you're going to mark them. And that's why you have on your lab kits, you've got marking pens. So there's permanent markers there. So that way, after the other person goes, you're going to give them all the marked ones, and you're going to wait a few minutes and they're going to go scatter those in the same area as your the remainder of the toothpicks that you didn't pick up, and they're going to send you out again. And this time you're going to do a recapture. And in the recapture, you're going to pick up as many toothpicks, and they can time you, as many toothpicks as you can find in that time, whether they have your mark or not. So marked ones and unmarked ones, you're going to pick them up. And then you're going to bring those back. You're going to count them. You're going to count how many you had in total. You're going to count how many you have with marks on them. You're going to mark the unmarked ones, give them back to the person. They're going to scatter them a second time, the marked ones, and then you're going to do a third capture. So you're actually going to be doing the initial capture, the first recapture, and then so we can do another technique, you're going to do a third recapture. So there's a video that explains all of this, so make sure you watch it. And then for doing the calculations, the video goes on to tell you how to set up your data tables and how to calculate the Peterson index, which is only going to be using your first capture and your second cap. So your first round where they put out all the toothpicks and you did a one capture, and then you marked them, they sent them out again, and you did a second capture. So that'll be the Peterson index, which is the most common index you're going to see talked about in papers. But then they're going to do it another time, and you're going to have the Schnabel index, because you can use all 3 rounds for calculating the Schnabel index. And you can see whether having the extra round of data is actually going to improve the accuracy, how close you are to the true population of 500 toothpicks in the estimate. So watch the video on how to set up this data table. Watch the video on how to do these calculations. And so we've got them all defined as the number that you capture, and the first time and the second time, divided by how many the ones you captured the second time had your marks on them. And that's going to give you an estimate of what the actual population size is. When you're done everything, you've done your data analysis, you've done your lab, update your blog, and then go to the assignment. You're going to submit your results and your spreadsheet, an Excel spreadsheet, just like you've been doing for all of the labs. You don't have to put your data into a form this time. If anybody, students want to see your data, they can go and look at your blog. So I will have posted a list of links to everyone's lab logs, and you can look at their lab logs as well. Now, at the end of the semester, you will be having it says a class data set will be posted. So I'll have to, maybe I will post a class data set. At the end of the semester, you're going to be having a lab exam. And just for you starting to think about preparing for that, these are some of the questions that you should be able to answer from doing this lab. And these are the types of questions that can appear on the lab exam. So the lab exam, you'll have all your labs available. You've got your lab book, you've got your blog with your labs, you've got all your Excel data sheets, you've got what we have on e-learning. And so these are the sorts of questions that you should be able to understand and answer. And on this particular assignment, how do your estimates compare to the true population size? And if they differ from the true value, why? Why didn't this work perfectly? So think about what some of the variables might be influencing this lab. Now, last week, I already went over how to write a lab report. We've talked about this a couple of times already. This week, I just want to talk specifically about Lab Report 1, and make sure that you do go over our Zooms from previous weeks. For your lab report, you must use class data from a lab that you have done. So you can't have skipped a lab and then write it up as a full lab report. It has to be a lab that you have done, and you have to use class data. So it's not just your personal data. And you're going to come up with a non-trivial hypothesis that can be tested. So a trivial hypothesis would be something like, I predict that there will be no birds in the bird survey, or that there will be lots of sparrows. Those are fairly observational things that we know you're going to find. So you want to come up with a hypothesis that maybe I want to look at bird surveys from forests in Florida and forests in New Jersey and forests in Maine. And I predict that the diversity will go down as I move further north, or go up as I move further north, or whatever. And you have to, when you are coming up with your hypothesis, so think about what is testable with the data that we have. You go into your e-learning. In the left menu, there's a point where you can click on that says labs. In there, it has all your instructions for how to write a lab report. There's a link to the video. There are the guidelines for lab reports, common mistakes, and a checklist. So the guidelines look like this. It's very detailed what goes into each section of your lab report. For some of you, this is the first time you've written a full lab report on your own. The guidelines will tell you what order to put the sections in. You label each section with a heading like introduction, methods, results, and discussion, and references cited. It tells you how to set up your citations, the in text and the end of the paper citations, and also how to set up your tables and figures. Now, if you're trying to get an idea for how it should look at the scientific papers that you're reviewing. Most of them use exactly this format. And also look at your textbook, because this is exactly the same format your textbook is using for citations and for tables and figures. Where do you put the captions? How do I refer to a table in the writing? So the sections, when your paper is done, when your report is done, are in this order: title, author, abstract, then the introduction, which includes your hypothesis, your methods, results, discussion, references cited. That is what I'm expecting to see. But that is not the order you will write them in. You'll put them in this order at the end. But the very first thing is you're going to look at some other papers. So you need to refer to at least 2 peer-reviewed scientific papers that relate to your hypothesis, that relate to what you're looking at. And then you're going to think about your hypothesis and take a look at the data you have. So start to analyze it, look at some tables and graphs, relate it to the hypothesis, and that way you get a feel for exactly what direction you're going. And so you've got the hypothesis written then, and then you'll have some tables and graphs. You can describe the data. Then you can go to the methods and you can explain how you got the data and how you got these results. So how I collected the pollination data, how I put together the class data, so who I got the data from, and then how I analyzed it. I took the number of pollinators and divided by the number of flowers to control for the number of flowers, or divided them into different color. Whatever it was you did, describe in detail your methods, and make sure you include where it was done, when it was done, what time of year, where did you get the data from. Then you can go, you've got your tables all described, you've got your methods, and you can discuss it. And so you discuss, did you reject your hypothesis, or were the results consistent with the hypothesis? We never prove a hypothesis because we don't know if the next study is going to disprove it. So we can just see, are we consistent with it, or have we actually rejected it? And often we use the papers that you read in the discussion or in the introduction. So you can also, which of these you write first is up to you, but the introduction is going to sort of set the stage for what your hypothesis was, where you were testing it, why you were testing it. And the discussion is, what did you find? Did you reject your hypothesis? Is it different than what you found in the papers? And what were some of the issues that you ran across? So once you've got all of that written, then you can make your references cited section, which are the full references in a APA format, and then write your abstract. So even though the abstract's at the beginning of the paper, you can't really write it till you've done everything else. So then you can write the abstract, come up with an informative title. Ecology Lab is not an informative title, but the effect of different colored flowers on bumblebees versus other pollinators is in New Jersey is a really good title. And then you're the author, and then proofread it, give it to somebody else to proofread for you, and that way you guys can review each other's and get some feedback before you actually hand it in. The video goes over all these sections of the paper in detail. So you can go through the video, watch the video on how to write a paper, and it includes going over these tips. So common mistakes I see on the first lab report: not having a clearly defined non-trivial hypothesis and not using class data. Those are the two big ones. And then sometimes when people write the methods, they just don't include enough detail. A lot of people leave out how they analyze the data. Not using peer-reviewed scientific references. So people will cite the textbook, the lab book, which is fine, but you still need 2 peer-reviewed scientific references. So websites are not going to count for that. And then not proofreading. Sometimes you leave things half said, or you've repeated yourself, or you've got something in the wrong section. And if you were proofreading, you'd probably catch all of that. Other common mistakes: dealing with tables and figures, not referring to them correctly, not labeling the axes on graphs, not writing full captions that explain what's going on in the table or figure. Some people sometimes will just take a screenshot of an Excel table, stick it in with no explanation. That's not going to work. Putting raw data in tables into the report. I don't need the full pollination data set. I need to see how you analyzed it. So what your results were. So I saw your raw data when you did your lab. I don't need to see it in this lab report. I just need to see the analyzed results. And some people will make a table which will have, the pollinators at different temperatures, and then they will show me a graph of exactly the same data. Just give me the graph. You don't need them both. Other common mistakes and titles that are not informative, like pollination lab is not very informative. Not fully discussing what your results mean and how it relates to other studies. And some people will just copy and paste the introduction or the discussion into the abstract, and it needs to sort of be pulled together and more coherent and talk about sort of a little bit of each section. Other common mistakes: there is a checklist on the guidelines. Use that. Make sure you're using the guidelines and the checklist. And then when you submit it online, it goes to Safe Assign. So don't email me your report. Don't send me a link to a Google Doc. Don't send it in a Pages format. So make sure that you submitted it correctly. You get an F in the course if you don't submit your 2 lab reports on time. So make sure you do get them in, because nobody wants an F. So tips: read peer-reviewed papers, use the checklist, start early. So start this week, not next. Don't plan on getting it all right in the first draft. Proofread it and don't miss the deadline. The first draft, when I grade it, I will give you some feedback. Students always do better on it. So the orange bars are the first draft grades, and then the yellow bars are the revised ones. And you can see the grades. This is the average grades improving, and the percentage of people who passed it, the first draft versus the second draft, increased drastically. These are a couple of examples from some past lab reports that students have written. Here's an abstract, so you can see they talk about their hypothesis. They talk about what data they got, what specifically the data were that they were addressing, what the results were, and how it related to their hypothesis. Here's an introduction for a different one. This is one from a MARC Recapture Lab. And here they sort of talk about the background to this lab, in which they use the textbook and they use the lab book, but they've also referenced of other papers that deal with MARC Recapture to discuss. They're comparing the 2 different models, the Lincoln-Peterson and the Schnabel models. So this week you got your exam on the Tuesday. You're going to start working on chapter 7 and then start working on your first lab report. Don't forget to do your MARC Recapture Lab. And every week you're going to be updating your scientific paper. And that's all I have. So hopefully you do well on your exam and can concentrate on something other than preparing for the exam this week. So take care.